And we are recording. So again, like I said, for those of you currently online, the first word for attendance is poster. And again, if you're currently online, that'll be an extra credit word. If you're watching this video at a later date, that's just your first attendance word, poster. Morning. When did you send it?
Last night at some point. This one was at
No, I haven't seen it yet then.
6. It was about so I tried getting the thing done once you get this an extension.
Uh-huh.
And I don't know if it was having anybody else, but it said that I had a private browser or something like that. And I tried getting through Chrome.
Uh-huh.
And I sent you a picture of it too.
It's interesting. Well, it's good that you took a picture.
Yeah, I mean, I had something like that happened to me before, and I didn't take a picture.
So is it when you were trying to watch the video?
It was just whenever I was trying to access the remote.
Really?
Yeah. So I mean, I did turn it in last night, so it is done.
Okay, that is really weird. I don't know what's up with that. Yeah, as far as I know, no one else has had that problem. If they have, they haven't told me. But I'm glad you. How'd you figure it out or fix it?
I don't know. I just wondered if it was working. So.
Huh. Yeah, maybe it was just some temporary glitch.
Yeah.
That stinks. So I'm glad you got it figured out. I got some more people here early. All right, so let's do a word. So it'll be extra credit, obviously, because it's 10 minutes before we're even supposed to start. So the next word will be stone. Like not a rock, not a pebble, not a boulder, but a stone. So let's take a look here. Burton is online. Cook is online. Hunter is online. Hannah is online. Cooper is online. Fisher was online. Hughes is online. Oof, not that there's anything wrong with it, but is there something going on? Not usually this many people online. Everybody okay? Anything going on I don't know about? I would have been online if I could. Because I also feel sick. Speaking of which, I mean, I'm going to repeat myself later anyway, but my office hours will be online today. So if you need me, I will not be in my office after class. Because like I said, I'm feeling under the weather. Lab will still go on as usual. The TA will handle it. Good question. So if anybody is sick, for labs, obviously labs are one of those things that you have to attend. But if you're sick and can get a doctor's written excuse or whatever, then you could just do the lab and get the data from somebody else and still get full credit if it's an excused absence, But again, you would need some sort of documentation. Like I think we had somebody earlier in the semester that had to miss because of a funeral. Maybe that was last semester. Anyway, they just provided documentation and that was that. And then they did the lab. But anyway. Excuse me. See, we got Crowder online. Galloway is online. Morning. Thought you were snapping at first. Like, oh, is everything all right? That was a. Julia is online. And just as a reminder, just because you're online, don't forget you still have to send the words.
Did you see the comment that I left from one of my independent works? You said I had like 90 points.
Oh, yeah, I fixed it. Yeah, you're good. Thank you.
I'll just make it
Yep, good call. Lewis is online. I'm telling you, there's a lot of people online today, and I'm jealous. Like I said, that's what I would be doing right now. If I could. Morning. All right, we've got about a minute left. I'll officially start attendance. If you're online, obviously, if I say your name and say you're not here, don't worry about it because you're not physically here, but just send me the words at the end of the lecture. So Abigail is not here. Kincaid is here in WVSU gear. Andrea, not here. Mullins, not here. Lewis is online. Cooper is online. Hunter is online. Milton is here. Crowder is online. Cody is here. Isabel is not here. Julia is online. Talbert's not here. Kennedy's not here. Hannah's online. Larissa is not here. Madeline is here. Hughes is online. Galloway is online. Cook is online. Durgan is here, right? Durgan? Yep. Tate is here. Burton's online. Kinder is not here. Jones is not here. Canterbury is here. Hobson is not here. Fisher is online. And we have Abigail and Kennedy are now here. And Andrea, right, is here. All right, it's 8. I'll wait till 8:01. Anybody I miss as far as names in person? Okay. We'll wait till 8:01. Well, in the meantime, like I mentioned earlier, I'm under the weather today, so I will be doing office hours online. So if you need me, meet me online for office hours. I will not be in my office. Lab will still go on as usual, except it'll be the TA by himself without my help. Larissa is here. And Zach is here too. And now it's 8:01 and everybody's accounted for. So again, please forgive me for my sluggishness today. Like I said, I'm not feeling good. Anyway, here's where we left off. Of course, if you already had lab this week, these words are not new to you. Actually, let's do attendance words first. That's the first one, the one I just underlined. This is the first word for attendance. Anyway, so yeah, these are terms that you are familiar with from lab. This is where we left off. Phenotype, genotype, those are definitely words you need to understand. You need to know the definitions of them. You need to know how they're related to each other. I might ask you the definitions, but more than likely I won't. Most likely there's words that just be used in a question. Like, you know, if a person with this genotype mated with this genotype, what are the chances of the child having this phenotype, for example? Anyway, so yes, we know, hopefully everybody's done the pre-lab by now and you're familiar with this. But the genotype, those are the genes you have, right? So, you know, like we mentioned on Monday, there's different versions of different genes, right? So for any given gene, there are different versions, and we call those alleles, right? And that's what your genotype is, right? It tells us what kind of allele. Specifically in lab and in this course, we just talk about genotypes that have, you know, one of two different alleles, right? So like we mentioned on Monday, the dominant allele and the recessive allele, right? So the dominant allele is always the one that masks the recessive allele. And that brings us to phenotype, which again, you should already know from lab. But phenotype is basically the result of the genotype. Now, your book in this portion of the chapter does define phenotype as a physical appearance, and I'll stick with that. But it's not necessarily always true, right? A phenotype is not necessarily always a physical appearance thing. Like somebody wrote a independent work paper about what was it, the P53 gene, right? So there was the normal allele, and then there was a non-normal allele, and that would cause possibly cancer, right? And that's not a physical appearance thing, but that is still a phenotype. But we'll come back to that concept when we get there. That's where we left off. Here's the new stuff. The next slide I'm going to show you shows a pair of homologous chromosomes. So this is not the first time you've been introduced to homologous chromosomes. That was during the meiosis discussion, but I told you they'd be back, and here they are again. In this chapter, we define homologous chromosomes as chromosomes that have the same, alleles of the same genes, right? So again, like we mentioned earlier in the last chapter, for all 23 of your chromosomes, you have two different versions, right? You got one from your mom, one from your dad. Those are your homologous chromosomes. If the gene for smelly breath is in the middle of chromosome number 13 in my genome, then it would be in the middle of chromosome 13 for your genome. I'm just making that example up. But the point is, that's what a homologous chromosome is, right? All the same genes. So for all humans who don't have anything wrong with them, all the genes are the same position on the same chromosome. Which brings us to this next word, gene locus. That is the specific location of a gene on a chromosome. So when I said, if the gene for stinky breath was on the middle of chromosome 13, I think that's what I said, that would be me describing the gene locus, right? I told you which chromosome number it was on. I told you where on the chromosome. I said towards the middle, right? That is what a gene locus is. And again, like I mentioned up at this first main bullet point, the alleles of a gene reside at the same locus on homologous chromosomes. So again, generally speaking, for all human, and I'm just using humans as an example because we're humans, but for all human genes, they're going to be in the same place, on the same chromosome for all humans, unless there's something wrong. And of course, as we mentioned earlier when we talked about the genotypes, right, whether you're homozygous dominant, heterozygous, homozygous recessive, that's basically just saying that right there, right? You can have identical alleles if you're homozygous dominant or homozygous recessive, or you can have different ones, which is heterozygous. So any questions about this slide? Okay. So here's what we're talking about, right? Here's a pair of homologous chromosomes, hypothetical ones. I don't know which ones they're supposed to be. It doesn't matter. We got one from your mom, one from your dad. And you can see here we're looking at three different genes. They're all on the same chromosome, but this particular loci, because that's plural, singular. You can see here we have, for that gene, big P, right? They happen to be homozygous recessive. So whichever chromosome we look at, they have homozygous dominant. They have a dominant allele. And you can see in the middle, homozygous recessive. Because no matter which chromosome we look at, they have a little a or little a. And then finally, here, for that B gene, whatever it may be, they're heterozygous because they're big B, little b. Those are just examples. I'm not going to ask you specific questions about this picture. But are there any questions about homozygous versus heterozygous? Okay. Let's talk about Mendel's law of independent assortment. This is also not a new topic. We talked about this in the last chapter, and I told you it was going to be back in this chapter. Here it is. But in this chapter, we're going to look at it from a slightly different point of view. And before we can talk about that, we have to talk about something called a dihybrid cross. So to remind you, previously we talked about a monohybrid cross. And in lab this week, you either did or will do a monohybrid cross, right? Crosses, because you mate with a partner, right? And you look to see, chin, dimple, or blue eyes or not, all those things, right? So you do a monohybrid cross. You do the Punnett square for that. A dihybrid cross, as you might imagine, is just two different traits, two different characters. So instead of just looking at chin dimple, for example, you might look at chin dimple in addition to, widow's peak. Of course, those are just examples. But anyway, if those genes for those two characters were inherited together. So again, in your mind, just think of any two characters in this lab, right? So the lab we did. If any of those two were inherited together, then an F1 would produce only the same two kinds of gametes they received from their parents. And the F2 would still show the 3:1 phenotypic ratio. There's no reason to write that down because that's a big if. I was saying if they were inherited together, like if you've got the the allele for dimple chin, then you also have the allele for widow's peak. And we know that's not the case, right? There was no situation where everybody who had a dimple chin also had a widow's peak, right? So that's not how it happens. What does happen, and this is what you need to know here, if the two characters are sorted independently, which they are, well, not always, but in this discussion they are, then the F1 would produce 4 different genotypes, not 2. And in this case, we're going to use the genes R and the genes Y as an example. So the 4 different genotypes would be, big R, big Y, little r, big Y, big R, little y, and then little r, little y. Right? So now, instead of saying there's a 50-50 chance like we did with the monohybrid cross, we're saying there's a 25, 25, 25, 25 chance, right? Equally. So because of that, the F2 generation, so to remind you, you've got the P generation, right? And then they have the F1 generation, which we just talked about. And then if you cross the F1 generation, then you would have the F2 generation. In this case, again, if we talk about the P plants, you would expect a 9:3 to 3:1 ratio. This is a ratio you should know and associate with a dihybrid cross. So the same way, when we talked about a monohybrid cross, we said there would be a 3:1 phenotype ratio, right? So using the example from Monday, we said there was about 75% purple flowers and 25% white flowers, right? That was a 3:1 ratio. Now, because we're dealing with more than just one character, we're not just looking at flower color, we're looking at 2 different things together. Because of that, there's a 9:3:1 ratio, and this is what it looks like. Pretty complicated, right? And that's only a dihybrid cross. Imagine something like a trihybrid cross, or even more than that. But anyway, yes. The long story short here is that alleles and genes, they don't segregate. They don't, how do I put it? They don't travel together always, right? So because of that, again, in this example, it's not like every pea plant that had a yellow pea, right, also had a wrinkly one, right? There's only one. Anyway, we'll come back to this concept. The more important concept, in my opinion, is the monohybrid cross that we talked about earlier. But this dihybrid cross that I just showed you, it supports the hypothesis, well, it was the hypothesis at the time, that each pair of alleles segregates independently of each other during gamete formation. So there we go. Finally, what you need to know about this topic. Each pair of alleles, I'll start right there. Yeah, that's what you need to know. Each pair of alleles segregates independently of other pairs during gamete formation. You need to know that for the exam. I will ask you the definition of independent assortment. So the inheritance of one character has no effect on the inheritance of another. And that is Mendel's law of independent assortment, right? So to give you an example, I'm going to try to draw this. This might work out, it might not. If you had, here's a homologous chromosomes. Actually, that's not going to work, is it? Stupid lines. Never mind. I'll show you a different picture later. But yes, know that definition of independent assortment. Now remember, when we talked about independent assortment in the context of meiosis, which is the first time we talked about it, it's kind of said the same thing, but we were concerned with it in a different way. Here, we're really looking at genes, right? So we're saying the same thing, but now we're focused on the genes. In the last discussion in chapter 8, what we said was that independent assortment meant, let me try this, I can draw. When your chromosomes line up, when your homologous chromosomes, oh, it kills me, line up in the middle during metaphase 1, right? It's not like all mom chromosomes are on one side and dad chromosomes are on the other, right? So for all 23 pairs of our chromosomes, it could be switched, right? They line up independent of each other. They have no bearing on each other, right? So using dogs as an example, this is not from your textbook. Again, if we're looking at a dihybrid cross, meaning we're looking at two different characters. In this example, the characters we're looking at are Black Coat and Vision. So Black Coat happens to be dominant. And by the way, if you're taking notes, don't feel like you need to study, memorize anything about these dogs. These are just examples. But anyway, Black Coat is dominant. Being able to see is dominant. Brown Coat is recessive, and being blind is recessive, right? So because of that, because of this dihybrid cross, we're looking at 4 different genotypes, right? Excuse me. Let's talk about the phenotypes. So this dog is black and can see, right? So we know its genotype must be big B something, big N something, right? And the reason we have the blank there is because this B could be a big B or a little b. Doesn't matter, right? Because that's the dominant trait. So all you need is one big dominant allele. Same with the N, right? That could be a big N there, could be a little N. It doesn't matter because as long as you have one big N, you're going to have that dominant feature, which in this case is the ability to see. And of course, you got this dog over here who has the dominant coat, right? The blind dog, dominant coat, but can't see, which is the recessive vision. So we know, again, to have the recessive condition, you would have to be homozygous recessive, right? Anyway, I'm not going to go through all these examples. These are just examples. Let's talk a little bit more about them before I get your questions. Again, this is looking at the same example. If you were to mate 2 dogs that were heterozygous for both, right? So they both are black. They both have good vision. If you were to mate those 2, and if they had a bunch of puppies, this is the phenotype ratio that you would expect. You would expect 9 of them to have both dominant phenotypes. You would expect 3 of them to have one dominant phenotype, and then recessive for the other. And then you would expect 3 more to have a different dominant trait and then recessive trait. And then finally, you would just have 1 out of all of those dogs that would have both recessive traits. It's a really long-winded way of saying what I've already told you, which is that you should expect a 9 to 3 to 3 1, 9 to 3 to 3 to 1 ratio when we're talking about a dihybrid cross. So any questions about this example? So again, to remind you, it's just an example. Don't study anything about these dogs and think, all right, there's not going to be any questions on the exam like, is black fur dominant or is brown fur dominant, right? These are just examples. The next word for attendance, right there, cross. So again, if you're online, make sure you send those words sometime before 9 a.m. To get your credit. And as always, or almost always, if you're in person, you can send those words too for extra credit. Whew, all right. So talking about those dogs again, let's use the test cross to determine an unknown genotype, because we had talked about that earlier. Well, let me back it up. This is an unknown genotype, right? If this is your dog, all we know is that it's a big B, and then we don't know if that second one's a big B or a little b. We don't, right? Because all we know is it has the dominant phenotype, and because of that, we know it has at least one dominant allele. Likewise, this one, right? It has the normal phenotype. It can see, so we know it has at least one dominant allele. We don't know what the other one is, right? And we kind of went through that today, or not today, but in lab. So how many of you, for example, have a widow's peak, right? So your genotype could be big W, or big W, little w. We don't know. I mean, we can figure it out by doing a Punnett square for your parents, but that's a discussion we'll talk about later in this chapter too. So how can we figure out an unknown genotype? That's what we're going to talk about. And the answer is a test cross. You should know what a test cross is. It's the mating between an individual of a dominant phenotype, but unknown genotype, and an individual that is homozygous recessive, right? So another way of saying that too is it's a mating between an individual of a dominant phenotype and a, recessive phenotype, right? So we know if it's a recessive phenotype that it has to be a homozygous recessive individual. I'm going to make sure I say that clearly before we move forward. I'll just use A as an example. Little a, little a. It could be any letters. To have a recessive phenotype, you have to be homozygous recessive. You have to have 100% of your alleles have to be recessive for you to have the recessive phenotype. Make sure you understand that. All it takes is one dominant allele, and then you would have the dominant phenotype, right? So you have to be homozygous recessive to have the recessive phenotype. Speaking of which, that is the answer to the pre-lab question that a lot of people missed. If two individuals who have a recessive phenotype were to mate, could their kids have a dominant allele, right? So if they have a recessive phenotype, that means the mom, and again, I'll just use A, would have to be little a, little a. They would have to be. To have the recessive phenotype, they would have to have the dominant, homozygous recessive genotype. Same thing with the dad. Would have to be little a to have that phenotype. I really hate this screen so much. Sorry. So that's supposed to be a Punnett square. I'm just going to stop. But yeah, so if you were to imagine doing a Punnett square, right? No matter what, 100% of those possibilities are all little a, right? So that's why, other than in the case of a mutation, which we haven't talked about yet, two parents with recessive phenotypes could not have a kid with a dominant phenotype. So a good example, no, that's not a good example. Never mind. All right. Anyway, going back to this idea of a test cross, right? So what we have here is this individual with a dominant phenotype. Therefore, we don't know the genotype. All we know is they have at least one dominant allele. We don't know what the other one is. So if we cross them, this is the test cross, with an individual who is showing the recessive phenotype, again, that means they would have to be homozygous recessive, then we could figure out what to expect, right? So these are basically two different hypotheses, right? We're crossing these dogs, and we're looking at two different hypotheses. If this dog here was big B, then we would expect 100% of their puppies to be black. And I wish, this is from your old textbook, I really wish they did a better job of this, because they only did the one square. Seems pretty lazy. They could just show the whole Punnett square to show you what it looks like. But yeah, no matter what, try to just picture it. If you cross the big B, or do it yourself if you want, with a little b, no matter what, 100% of those offspring are going to be heterozygous. Therefore, they're all going to show the black phenotype, right? So again, if you were to cross, this is a test cross, if you were to cross these dogs and 100% of the puppies were black, that would be an indication that that dog is probably homozygous dominant. It's an indication, it's not proof. Meanwhile, if this black dog was not homozygous dominant, if it was heterozygous instead, and again, I wish the old textbook did the whole Punnett square to show you, but in that case, and you can do it yourself if you want to do a Punnett square with big B, little b, cross with little b, you would see that 50% of those puppies would be heterozygous, meaning they would be showing the dominant phenotype. And you would see that about 50% of the offspring should be brown, right? Show that recessive phenotype. Does all that make sense? So again, critical thinking here, because dogs and puppies and colors is not really important. But think about this. What if you cross these dogs like we talked about, and 100% of the puppies, let's say you had 5 puppies, 100% of the puppies were black. Is that proof that this dog here is big B if 100% of the puppies are black? That's okay. The answer is no. It's not proof. It's pretty good indication, right? Because what we would expect is 100% black puppies. But it still is possible that this mating right here on the right, we would expect 50% black and 50% brown. But there is a possibility that 100% of them could come out black, right? This is all probabilities. And we'll come back to that concept here in a second too. But are there any questions about this test cross? It's just an example. All right, like I said, the rules of probability. So that's what we're going to jump into right now. Like, what are the chances of this versus that? There's going to be a few questions on the exam where I do say that. I'm going to say a parent who has this mates with a parent who has that. What are the chances that their kids are going to have this? Or how many of their offspring would we expect to have that? So I'll give you some examples later. But genetic crosses do obey the rules of probability. And these are the same rules that apply when you toss a coin, roll the dice, draw cards, all that kind of stuff, right? So when you toss a coin, the 50% chance is going to be head, 50% chance is going to be tails, right? When you roll the dice, if you were to just roll one, there's a 1 in 6 chance of it landing on any particular number, 1 through 6. But that brings us to what you do need to know, which is the rules of multiplication, which states that the probability of a compound event is the product of the separate probabilities of independent events. This is a long-winded way of saying this. You just multiply the chances, right? So what are the chances of getting ahead? All right, let's say I'm flipping a coin and, I don't know, he's flipping a coin. And we say, all right, what are the chances of me landing, flipping and landing on head, and him flipping and landing on head? Both at the same time, right? So, well, the chances of me doing it are 1 and 2, right? The chances of him doing it are also 1 and 2. So the chances of us both doing it at the same time, again, you multiply it. So this is what you need to know for the exam. And that gives you 1/4. Those are your chances. So for example, on the exam, when I give you some kind of question, it might say something like, all right, there's a 1 in 7 chance of this couple having kids with this particular disease, right? And so I might ask you, so what are the chances of this couple having a kid with the disease who's also a boy? So like I said, 1 in 7 chance of having whatever hypothetical disease I'm talking about. There's a 1 in 2 chance of having a boy over a girl. So what are the chances of having a boy with that disease? You multiply it and you have 1/14. So yes, there will be a little bit of math on this exam, but it will be very, very basic like that. Very basic. The numerator is always going to be a 1, so you don't have to worry about that. The denominator is going to be some easy numbers to multiply. They're not going to be fractions or anything extremely large. Yeah. So again, you will get questions on the exam where I give you these hypothetical situations. Say, all right, what are the chances of this and this and this? So make sure you need to understand this rule of probability. So any questions about the rules of probability? All right. I'm working on an online lab for that. If I can get it done in time, I'll share it with you guys if you want to do it for extra credit. I mean, I'm sure no one wants to, but if you decide to do it for extra credit. So let's talk about family pedigrees. So, speaking of which, this kind of comes back into play. You'll see why later. But Mendel's principles that we've been talking about when we talk about these pea plants, right? Whether it be pea color or flower color or whatever, all these things we've been talking about, they also apply to the inheritance of many human traits, like a lot of the ones that we talked about in lab, for example. Your old textbook gives these examples, 2 of which we looked at in lab, 1 of which we didn't. Actually, I might add freckles to it for next semester. Anyway, so freckles are dominant, not having freckles is recessive. Widow's peak is dominant, not having it is recessive. Free earlobes are dominant, attached earlobes are recessive, right? These are just examples. Of course, now this is the second time you've seen these examples, both in here and in lab. But still, I want to remind you, these are just examples. I'm certainly not going to ask you any questions like, is a widow's peak dominant or recessive? Who cares? None of that's important. Freckles are not important. Attached earlobes are not important. All the stuff we did in lab, those particular examples are not important. It's just good examples, right? Just good practice, being able to do a Punnett square and be able to understand this relationship between dominant and recessive. But anyway, back to this whole idea of a family pedigree. Dominant traits do not imply that they're normal or that they are more normal than common. So for example, again, let me think of a good one from lab. Oh, we did that one. Does anybody remember off the top of your head which traits were there like more? We had more recessive than dominant. Does anybody remember? I don't remember off the top of my head, but cleft chin? Yeah, that's a good one. So raise your hand if you have a cleft chin. Right? No one in this right now. Someone online might. The cleft chin is dominant, yet no one in this classroom has it. Right? So again, dominant does not necessarily mean more common. Again, that's a great example. So thank you for that. And what we call that, and you need to know this for the exam, is it's called a wild type trait. The wild type trait is the one that's more common. And had I not left early on lab on Monday, we would have done that part that you guys didn't do, which everybody stands up and we have everybody sit down if they don't have a certain feature, because we wanted to see how long it took for everybody to be unique. So those are some of the things that we would have seen in lab. But yes, again, dominant does not necessarily mean more common. And then one more example, because we just saw this on the previous slide. Raise your hand if you have freckles. Okay, 3. So again, a very small portion of the people in this room have freckles, but freckles are dominant, right? So in this case, non-freckles would be the wild type, at least in this samples that we just did. Cleft chin, or excuse me, smooth chin would be the wild type trait. So any questions about that topic? All right. Anyway, back to this idea of family pedigrees. How can human genetics be studied, right? So we've been talking about Mendel and what he did with pea plants. We can't do that with humans, right? So he would cut the male parts off and make this flower mate with that flower, right? We can't do that with humans. We can't be like, I wonder what this would be like. I need you to have sex with that person so we can study your children and make your children have sex with those, right? It's unethical. We can't do that. So how do we do it? Well, we have a pedigree, right? Geneticists analyze the results of matings that have already occurred, right? So it's unethical to make humans mate, but we can look at matings that have already occurred. And that's what a pedigree is, right? A pedigree is basically a family tree. A family tree can be used to determine if a trait is sex-linked. I'll give you an example of that a little bit later too. The next slide I'm going to show you, I think it's the next slide, shows the pedigree tracing of the incidence of a free versus attached earlobe. So again, not important, this whole earlobe thing. It's just an example. And if we use Mendel's laws, we can deduce genotypes. And let me show you what I mean by this. Bear with me. So here we go. If the color is purple instead of white, that means they have the attached earlobes, right? So it's a male, square, female, circle. In this discussion, male versus female doesn't really matter. The idea here is, do they have the dominant phenotype or the recessive? So again, purple is recessive phenotype. So I'll go ahead and write those genotypes in, right? We know those genotypes have to be little e, because those are the recessive phenotypes. Is everybody with me so far on that? Hopefully that makes sense. For them to have the recessive phenotype, they have to have the homozygous recessive genotype. So there we go. Now, how about, and we're not going to do all this. We don't have time for this, but I'll hopefully give you some more of these, and you can do them for practice for extra credit if you want. But let's talk about this person here. Let's figure out what their genotype is. So, what should the first letter be, big E or little e, for this person? I think someone said it. Big E. No matter what, they have to have at least one big E, because they don't, they have the dominant phenotype, right? So we know they at least have one dominant allele. How about the next one? That's where it gets a little bit trickier, but we can figure it out. What must that next letter be? Big E or little e? I like that, because when we're just talking about this person, it could be a big E or a little e, right? If we're just talking about this person, but we're looking at a bigger picture. These two people had these kids. So what must that person's genotype be? Think about it this way. This one, who got all their alleles from their parents, has little e, right? So that person must be big E, little e. Because if they were big E, and that's the female, when they produce eggs, 50% would be big E, and 50%, the other 50% would also be big E. So no matter what, if this person was big E, even with crossing with that person, their kids would be heterozygous, right? Do a Punnett square if you need to see what I'm saying. But yes, if you were to cross big E, and big E, little e, 100% of the offspring would be heterozygous. So the only way to have a homozygous recessive offspring is for each of the parents to at least have one recessive allele. And I'll leave it at that. We could do that for the rest, but I don't want to waste your time. It's the same concept. It really is good practice. I really doubt any of you will ever do a pedigree in your life. You're not biology majors, but if you do this, and if I can give you some more for extra credit, it is good practice because it really helps you understand this idea of heredity. And more importantly, in my mind, it's critical thinking, right? This is good ways to figure things out, right? Figuring out that person has to be heterozygous, otherwise they wouldn't have any homozygous recessive children. So anyway, yeah, keep an eye out for that. I'll try to give you some practice you can do for independent work if you want, or extra credit. I don't know yet. Let me think about that. Anyway, let's talk about human disorders that are caused by a single gene. And gene will be the next word for attendance. Human disorders caused by a single gene. Some of them are known to be this way, where it's dominant or recessive, right? The next slide I'm going to show you is going to show a table of some of them. And these are what we're talking about. You do not need to study this. As a matter of fact, I'm not even sure if this one's from your new textbook or your old one. I think it's from your old textbook. But these are just examples. You definitely don't need to know them, right? So these are some examples of recessive disorders. These are some examples of dominant disorders. And in a sense, they're basically, at least the way we're talking about it, the word disorder could be substituted for phenotype. These are just phenotypes in a sense, right? These are dominant phenotypes. Those are recessive phenotypes. And we'll talk about some of them here in a second. But again, when you're studying for the exam, please don't feel like you need to memorize any of this. I'm not going to have any questions about cystic fibrosis or PKU or achondroplasia. Well, I might. But if I do, I'll give you all the information that you need to know, right? So again, there's no need to study and memorize any of this stuff. If there's any questions about any of these things, I will give you all the necessary information you need to know to answer it. All right, so let's talk about recessive disorders. Most of them are recessive. Most lethal human disorders are recessive, and you'll see why shortly. But for now, just keep that in the back of your mind. Maybe think about it. Maybe I'll ask you later. Individuals who have recessive alleles that appear normal are called carriers. So again, this is the same stuff we talked about earlier, but now we're just talking about it in different contexts, so we're just using different names. In this case, carrier, yeah, well, what do we call that person before? That would be somebody who's heterozygous. And I'll just use T as an example. Big T, little t, whatever letter. An individual who has a recessive allele that appears normal is a carrier. And again, that is something that we would have called a heterozygote person previously. And they are. It's just a different way of saying the same thing. With the stuff we learned earlier, we can predict a fraction of affected offspring that are likely to result from the mating between two carriers, which is, again, there's going to be some questions like that on the exam, which is where I say, what are the chances of this person mating with this person and this being the results? Those are the kind of things you're going to need to do for the exam. So here's an example. And again, just an example. You don't need to memorize anything from here. If we have two normal people, when it comes to hearing, they both can hear just fine, but they're both carriers, right? So they're both heterozygous. If they were to mate, what are the chances of them having a kid who has normal hearing and is not a carrier, right? Well, if you were to do the Punnett square, you would see there's a 1 in 4 chance of having a kid that has normal hearing that's not a carrier. What are the chances of them having a kid who has normal hearing but is a carrier, right? That's, 2 to 1. Well, 2 to 2, right? 50% chance of that happening. What are the chances of having a kid who's normal in hearing? Well, then you'd also consider that one, right? So you could say 3 to 1, right? What are the chances of having a kid that's deaf? 1 in 4. So yeah, these are the kind of questions you might get on the exam. And again, this is just an example, the hearing thing. So in the exam, you would get something like where I would say, hearing loss is a recessive disorder. It's not necessarily, it's just an example, but hearing loss is a recessive disorder. What are the chances of a carrier mom and a carrier dad having a boy who can hear? Well, you would do a Punnett square like that and figure out, okay, well, there's a 1 in 4 chance that, 3-to-1 chance that the kid would be able to hear, as we can see from here. And then I said, what if it's a boy, right? So 1 in 2 chance of that happening. What I say? 3 to 4 chance of the other one happening. So you multiply that out. You go 1 times 3 is 3. 2 times 4 is 8. So there's a 3 in 8 chance that they would have a boy who can hear. That's just an example. So again, don't memorize that math that I just showed you. That's not a problem. Those are not the exact numbers I'm going to give you on the exam, but it could be something like that. Any questions about this example before we look at different possibilities? Because again, these parents are just both carriers. No, never mind. That's the only one, my only example. Anyway, cystic fibrosis. This is the most common lethal genetic disease in the United States, which is, I figure, why your book talks about it, right? Because there's a bunch of them. I just showed you the table, but this one is the most common. It's caused by a recessive allele that is carried by about 1 in 31 Americans. Let me put a big old X through this. The only reason I'm even talking about this is because your book did, and I teach out of the textbook. I'm not going to ask you anything about cystic fibrosis. You don't need to memorize anything about cystic fibrosis. It's just an example of a recessive disorder. Whew. So any questions about recessive disorders? Let's talk about dominant disorders. Some disorders are caused by dominant alleles, and achondroplasia is a great example of that. Achondroplasia is a form of dwarfism. But this one's an interesting situation. Again, this is just an example. There's no need to memorize what I'm about to tell you. If I have any questions about achondroplasia on the exam, I will give you all this pertinent information. But anyway, the reason this one's interesting, well, first of all, let me back up. Again, it's a dominant disorder. And like I said previously, when we were talking about recessive disorders, you could also use the word phenotype. It's the same thing. It's a dominant phenotype, meaning for these disorders, if all you need is the one dominant allele, and then you're going to have that disorder, aka have that phenotype. That being said, achondroplasia is a little bit different. So yes, you still need the one allele to have dwarfism, because it's a dominant disorder. But what makes this one a little bit more interesting is that you cannot have anybody who is homozygous dominant. It just so happens that that's deadly. So every dwarf you see with achondroplasia has to be heterozygous. This is a dominant disorder, so we know they have at least one dominant allele. But we also know if they were to have 2 dominant alleles, in this case, it would be lethal, and they just wouldn't exist. So it is an interesting example. Again, I teach out of the textbook. If it were me, I don't know, I might not even mention this in the textbook, because to me that just kind of confuses it. It's like, hey, this is how dominant disorders work. And then I'm like, except this one. This one's different than the rest. But yeah, just remember, a dominant disorder, as far as we're concerned, is the same as a dominant phenotype, right? All you need is that one dominant allele, and you're going to show the disorder. You're going to show the phenotype. So here's an example of, again, some crossing. So we have a normal parent crossed with a dwarf parent. What are the chances of them having normal kids? 50%. You can do the Punnett square yourself and see that. What are the chances of having dwarf children? 50%. So these are the kind of questions you might get in the exam. And again, I would remind you, because I don't want you to memorize this, but I would remind you of this whole thing about homozygous dominant being deadly. But yeah, and it might not be dwarfism. I could ask you about anything. I might make up some disorder, but if I say it's a dominant disorder, then you should know how this is how it works. You should be able to do a Punnett square. So again, normal parents, normal parent crossed with a dwarf parent. Those are the results. Actually, yeah. And if you wanted to, I'll take care of this later. But I really would like to come up with some examples that you could do on your own for extra credit. So like, for example, a dwarf crossed with a normal person is what we see here. What about a dwarf crossed with a dwarf? What are the chances of them having a dwarf child, or the chances of having a normal child, or the chances of the child dying before it's even born? Homozygous dominant thing. Anyway, any questions about this? All right. Dominant lethal disorders are much less common than lethal recessive disorders. I mentioned that earlier when we were talking about recessive disorders. I said they are more common than dominant disorders. And hopefully that makes sense to you if you think about it. Because with a recessive disorder, you have to have both alleles to get it, right? So it's very possible You could have it, you could be a carrier and survive and have kids and then pass along that bad allele, right? But with a dominant disorder, with some exceptions, if you have that one allele, you're dead, right? If it's deadly, right? So that's why dominant lethal disorders are less common, because people usually die before they can reproduce. So even though, yeah, the chances of them passing along that allele would be very high, the problem is they're not surviving to do it. And there's the exception to the rule, which is Huntington's disease. So I'm not going to ask you any questions about this, but long story short, and you can read about it if you want, it's just an example. Long story short, Huntington's disease is fatal, but it doesn't kill you until later in life. So what I said about dominant disorders versus lethal disorders, it's still true, but it's said in this case, that person can survive, and therefore they can survive to an age where they're old enough to have children. Therefore, they could pass along the disease, right? So again, normally these lethal dominant disorders are less common because they kill you, so you can't pass along your alleles. But in some cases, you can survive long enough to do that. Anyway, any questions about Huntington's disease or this idea of dominant disorders? All right. I left this in. This is from your old textbook. The old textbook. Every chapter had something called the process of science, which is where they basically talk about a particular science experiment. I'm certainly not going to ask you about this. It's not even in your textbook. But if you download the PowerPoint, you can look at it if you want. You can read about it. And if you wanted to, you could do a research paper, independent work paper on it. Anyway, we've talked about recessive disorders. We've talked about dominant disorders. Let's talk quickly about genetic testing, since we're talking about disorders. We have tests that do detect disease-causing alleles. So all these disorders we've talked about, well, we have tests for them. And actually, let me go ahead and tell you this too. I'll put a big old X through this. I'm going to go through this quickly because I'm not going to ask you any questions about this. There might be some on the study guide, I don't remember. But I'm not going to ask you any questions about this. It's not a health class. So this is important information, I guess, but not important for us. So everything I'm about to teach you, if you brain dump it when you leave this room, you'll still be able to understand everything I teach you later in the semester, which is why I consider this relatively unimportant in that context. Anyway, some testing. There's something called amniocentesis. That's where they take some fluid out. They can check it that way. That's what they're doing. You can see they've got the needle there. See the pregnant lady. They're doing an ultrasound. You want to make sure you don't stick the baby with the needle, right? That's what it looks like. Then there's CVS. That's another way of doing it. You can read all about it if you want. I'm not going to test you about it. Yeah. 4 minutes. Yeah. Yeah, that's where we'll end. We'll end 4 minutes early. And I guess early will be the last word for attendance. And I will see you, I don't know. I might come to lab today, but it'll probably just be the TA. So I'll probably see you on Friday. I'm not feeling good. If you came in late, make sure you send me an email.